probability on like how frequently you'll find a restriction site? Is it just based on how many base pairs are in that restriction site? Like
So
if it's 6 long or 5 long or whatever?
Number 10, I added a wrinkle that we hadn't talked about in class yet. This is a little bit different than the normal, how frequently does this restriction site appear? So the question is the entire question just asking how many different nucleotides there are in the recognition sequence? Help me out. On one hand, the answer is yes. The number of nucleotides does matter. Why is that? The more nucleotides you're looking for a specific sequence of, the more frequently or less frequently you find it. Right. If you're looking for a random sequence of nucleotides, it's more likely you find a smaller sequence more frequently. So if I say this restriction site recognizes the palindrome AT by themselves together on the chromosome happen quite frequently. Like here's one in question 11. So if you look for a combination of 2 nucleotides, then that is, the probability is at every site you're looking for this is the trick, not a trick. Trick is my favorite word. The concept is at every site, when you're looking for a specific nucleotide, the probability is 1 in 4. If you're looking for an A or a T or a G or a C in a random DNA sequence, if you close your eyes and point at a nucleotide, oh, I almost got one. The probability is 1/4 that I pointed at a G. Otherwise, it would have been an A or a C or a T. Right? So 25%, 1/4 chance of one nucleotide. So that could be that C, for example. And then you have to figure out what the probability of the next nucleotide also being a C is. And that's also a 25% chance. So the combination here is you just have to remember to multiply. And if you don't remember on the exam, then you can always come back and look at this video and remember. Just don't listen to the audio. You can watch the video if you want. That you multiply the individual probabilities together to come up with the total probability. So on one hand, the calculation does have to do with the number of nucleotides you're looking for, because there's going to be one of these multiplication terms for every nucleotide in the recognition sequence. But
oh, I was going to ask, would you accept a percent instead of a fraction?
Yeah. You just have to, the trick there is that you have to know how, this is asking how frequently. Which I guess I would consider percent to be a reasonable answer to that question. So getting back to this example, what have I changed here compared to everything else we've done before? There's something new. There's a new nucleotide. What's the 5th nucleotide in this sequence? N. What does N stand for? Any. So what's the probability of finding any nucleotide wherever my finger lands on a random piece of chromosome? 1. There's a nucleotide there. It's any nucleotide. So the probability of that term, oh, I should add a third 1/4 here. So the first 3 nucleotides, CCT, the probability of each of those is 1/4. C, quarter times 1/4 C times 1/4 T. 1/4 times 1/4 times 1/4. And then we get a string of 5 N's. Probability of any nucleotide at those 5 positions is 1. So that makes it times 1 times 1 times 1 times 1 times 1. Those 5 nucleotides, you're guaranteed that any nucleotide will be at those 5 positions. So then we have to get back into, after that, the AGG. What's its probability? That's like the first bit, 1/4 times 1/4 times 1/4. So the probability of getting an A is 1/4, G is 1/4, G is 1/4. So it's if you want to simplify it, if you want to reduce it, if you're like that, It's a quarter to the 6th. It's a quarter times itself 5 more times. Quarter times a 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 quarter, which is whatever I wrote in the answer key. You can plug into Google Sheets or Excel or a calculator and get the answer. So yes, the frequency depends on the number and the size, the length of the recognition site, but in this case, the n's basically don't do anything to the math. And those n's don't have to be in the middle. They could be anywhere in the sequence. They would still have the same effect, that you would just multiply whatever your answer was by 1. So I could add an n, I could add 3 n's onto the end of that sequence, and it would still be the same probability, 1/4 to the 6th. Does anybody want to know what the answer actually is, instead of leaving it as a fraction? Good. We could do it in our heads, probably. Yeah.
Isn't there another letter nucleotide we have to know for the exam?
You don't even have to know n. I mean, I did describe it here, but what are the other ones? Did we talk about another one?
I think one of them's like a Y or something.
Oh, well,
S and W.
yes. We did talk about S's and W's. So we might as well review that. That's quick. S stands for strong. Those are the nucleotides that make 3 hydrogen bonds. So an S nucleotide would be a G or a C. And weak, W, would be a nucleotide that's either A or T. So you could have, for example, a recognition sequence that was, I'm making this up. You maybe couldn't have a recognition sequence that was strong, strong, weak, weak. That would be a palindrome, wouldn't it? No, it wouldn't. But anyway, just for the sake of example, let's say that was a recognition sequence. What's the probability of getting at one spot a strong nucleotide? S stands for G or C, so that's half of the nucleotides. Of A, T, G, and C, there are 2 that are strong, G and C. So the probability of strong would be a half, because that could either be G or C. You've got a 1 in 2 chance of picking one of those at random. And then, again, half. And then the weak, A or T, you've got a 2 out of 4 nucleotide chance of finding a weak nucleotide there and a weak nucleotide next to it. So that would be half, fourth, eighth, 1 in 16. So the conclusion here is 1 in every 16 groups of 4 nucleotides, you would find one that was strong, strong, weak, weak, on average. And there is also 2-letter code for purines and pyrimidines, but we didn't talk about that in class.
Is it 1 in 16 individual bases or like groups of 4?
You can think of it in one of 2 ways. Number 9 that I wrote all over. Let's see. So I'll need somebody to tell me what the DNA sequence is that BAM-H1 recognizes. I bet it's a palindrome, and there's probably only one in here.
It's the GGATCC.
Okay. So in that case, I definitely expect you to get on your favorite web browser and Google BAM-H1 and find out what recognition site it recognizes. The other thing that we need to know is where does that endonuclease cut the strands inside that 6-base region. So now I've got to zoom into the full sequence. So we can write in base pairing, Hopefully I'm doing this properly. Somebody fact check me while I'm going.
You're good.
Awesome. I love validation.
Oh.
It's kind of like when you go to the store, if you've got a debit card or a credit card, and at the end it says you're approved. It's like you get this nice happy glow. All right. That should work. Almost perfectly lined up. Okay, so did somebody find out where this enzyme cuts GGATCC? Okay, so I heard between the two Gs. So that enzyme is going to bind to the DNA, and it's going to cut the backbones in between the two pairs of Gs. So then all you have to do, although this is not at all clear from the outset, is rewrite those. So I'm going to cartoon now. The top strand to that point, and then there was a break. And then it continues on that side. So this is representing then the bold sequence up here. There's that much DNA sequence, then the restriction enzyme cut the backbone, so you're going to get two fragments of DNA from one strand. This left fragment and the right fragment. I'm just not writing in the letters. So on the bottom strand, that is going to be longer because this enzyme makes a staggered cut. So on the bottom strand, it cut between those two Gs and then continues along the chromosome. And at present, these molecules are still base paired with each other. Like this. But the backbones have been cut. So the question really is, what would happen if you heated this chromosome up a little bit and the chromosomes melted? The hydrogen bonds broke. At some point, you might wind up with something that looks like those two pieces of DNA are separate like this. And this is really important later when we start talking about cloning using restriction enzymes to recombine different pieces of DNA. We can cut chromosomes and take pieces out of the chromosomes. We use the restriction enzymes to break the chromosomes. Sometimes they leave these overhangs that are no longer base paired with each other. Those are sticky ends. You don't need to know that terminology yet, but we'll get there. So the hydrogen bond donors and receptors are hanging out in the nucleus, but they're not necessarily base paired with each other. So not all restriction enzymes make these staggered cuts, where you get these weird ends hanging out by themselves. Some enzymes make blunt cuts, where they cut the backbone immediately across, same point in the backbone on both strands. You get blunt ends, where there's no overhang of one strand. What about DNA polarity? Does anybody have any questions about this one on labeling the polarity of those products? If that's the 5' end right here, then what's the nucleotide right here on this strand? It's another 5' end. So anytime there's a break, this is kind of like DNA replication. This might have been a lagging strand that gets synthesized right up until that point. And then this would be the start of the next, if this was lagging strand, this would be a DNA or an RNA primer, and that would be its 5' end. So you can cut the backbone as many times as you want. You just make lots of shorter fragments that have the exact same polarity. They're all going to be 5' to 3' in the same direction. So left to right, 5' to 3', 5' to 3'. On the bottom strand, it's anti-parallel, so that would be 5', the 3' end of that fragment, and then another 5' and a 3' down there. and if you're concerned about that, just for completeness, we'll note that if that's the 3' end, that means there's a hydroxyl group. So that one nucleotide, whatever nucleotide that is, has a 5' phosphate. That's over here linked to the next nucleotide, and it ends in a 3' hydroxyl. And what's on the 5'? There's an atom there that's important for nucleotide phosphate. And that's why we know that's a 5' end. So we really zoomed in and looked at the chemical structure of that nucleotide right here. It has a free 5' phosphate linked to the 5' carbon in the nucleotide, and there's a hydroxyl right next to it. So in this case, the restriction enzyme is doing the opposite of what DNA ligase does. A restriction enzyme comes and breaks that chemical bond that separates the hydroxyl and the phosphate. Ligase does the opposite reaction. Ligase, at the end of synthesis, would come in and reconnect the oxygen and the phosphate, get rid of that hydrogen, and give you the phosphodiester covalently bound backbone. So those two enzymes do the opposite.
For this one, would you just want us to draw that, or do you want us to draw the base pairs?
You wouldn't need to draw the base pairs, yeah. Or, good testing strategy, diagram it first so that you've got something down, and then if you've got time at the end, if you really feel like you want to go right in the individual nucleotides, you could. Yeah, microsatellite notation, definitely. Anything we did exercises that involved good plan to study for it. Microsatellite notation we already saw a little bit today. How would you rewrite that in microsatellite notation? What's the repeat with the unit of the repeat? Is it TA or is it AT? They both have 4 repeats, so so there's no right answer. You could do either one. You could either say this is TA 4 times, and then there's a T, this one, that doesn't have an A following it, so it's not part of the repeat. Or you could call this T, and then AT4. If you come up against a question like this, the way you would distinguish which one to use is which one shows up more frequently. So if there was a situation where you could say there were 4 repeats of one thing or 5 repeats of something else, you'd want to pick the one that has the larger number of repeats. So for example, if I had added another A to that initial sequence, that would make it TA5. Otherwise, you would call this TAT4A, which isn't as concise. It's not as good a shorthand, so that would not be the way I would answer that question. If there's the same number of repeats, it doesn't matter. Yeah?
Could you do TA2?
Sure, if you wanted to. Yeah. Oh. Well, that's not, why not? I like creativity. So, yeah, you could say that as, although it wouldn't be as concise, you could say that was Tata 2. Or you haven't simplified, it's like reducing a denominator. You haven't simplified it as much as you can. I mean, if you really want to get crazy, you could call that TA2 2.
Wow.
No, don't do that. Don't do that. I mean, I would understand what you were trying to say, I think, but just stick with the simple stuff at the top.
What about TA 2.5?
Ooh. No, you could do that, but nobody in genetics actually uses decimal microsatellite repeat nomenclature, which you didn't know until you asked, but now I've told you. Don't use decimals. in the denominator, or in the subscript. Don't use decimals down there. Whole numbers, please. Only whole numbers. The first exam, I'd probably give it to you anyway. But I would write a note that said, please don't do this in the future. Anything else I can do to help calm fears, ease minds, aside from giving you the exam in advance or offering my help during the exam? By the way, so while you're thinking about that question, a couple of last things to think about, just reminders. Do please know how to take screenshots on whatever device you're going to use to do this so that you've got a backup. Because I've had students say that an app crashed or something during an exam, so I just want you to regularly please take screenshots just in case. Then you've got evidence on your photo roll on your device that you've been doing the work. After the exam, I do teach a class right after the exam, but at some point immediately after the exam, I'm going to quickly go through everybody's submissions and make sure I can see what you've submitted. Do check your email through the afternoon, because if I do have any issues, I will be emailing you and expecting a pretty rapid response if I need any additional information from you about what you've turned in. If you work after class, then please tell your manager or supervisor or coworkers that you might have to step away for a minute and finish something. It's very rare, but it could happen. When you come into class, the exam will be distributed on Google Classroom exactly at 1 o'clock, and then it'll be due by 1:50. So you've got 15 minutes to work on it. Bring written notes if you want. You can use notes that are on your device. It's open internet. So you can either, if you want to have paper copies of your notes, you could print out digital versions if you want to have them on the side, so you can have your paper notes in your device that you're working on. Just don't bring 2 devices, please. 1 phone, tablet, or laptop per person. You can bring a calculator if you want to. I don't see why or why not, but that's okay. And no interpersonal collaboration. That is, you can use the internet, but you can't seek or accept any real-time help from anybody else. No collaboration, in other words.
If we have technical difficulties, like in the middle of the exam, with one device, are we allowed to switch devices?
So if your device ran out of battery during the exam, then you could switch devices. You can bring more than one with you, but you can only use one at a time. Let's put it that way. Bring as many laptops as you want. But you can only use one at a time. If you have any other questions about exam, anything, email me, let me know. I've got office hours right before the exam. Well, 11 to noon, rather, on Wednesday, so I expect to see probably some people there as well. And I'll see you Wednesday. So if you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 nucleotides. The one way to think about this is you're looking at every set of 4. So there's a set of 4. We're asking, is that sequence in that set of 4? No. Move to the next group of 4. Is it one of those? No. Is it that group of 4? So you're basically asking every time I look for a set of 4 nucleotides, every 16th set of 4, you would be expecting to find that sequence. That's a relatively minor point. Not super likely to have a question that's related to that on the exam. But in function, that's what this is asking. Another way you could do the exact same thing, which would be equally as valid for completeness, was you could ask for every serial set of 4 nucleotides. 1 through 4, 5 through 8, 9 through 12, 13 through 16. So it could be overlapping or non-overlapping. Doesn't really matter. I would have to specify what I was looking for, basically. Overlapping or non-overlapping sets of 4. Other questions? So look at, in this case, 16 groups of 4 nucleotides. You'd expect 1 out of every 16 groups of 4.
need to know how to draw the egg and sperm and how like the DNA replicates, where we had the bolded line, the thin line.
So that aspect of synthesis, yes, but we didn't get into mitosis and meiosis. So I won't specifically be asking questions if they were to be on the exam about sperm and egg specifically. No meiosis, but the aspect of template strand and newly synthesized strand, yes. But once we started talking about mitosis and meiosis, that content won't be on the exam. You're a particularly reflective group. I like it. Although sometimes these things get way more animated and excited. So that's either a good thing or a bad thing. You're either very confident or not. But that's reasonable going into the first exam. You have seen an example exam now, but that doesn't necessarily help. Yeah.
Could you do number 9 on that?
I definitely will. Number 9.
It's the one where you're multiplying the 1/4